Respiration is the overall process of catabolism, from the breakdown of a substrate, such as glucose, to the reduction of a terminal electron acceptor, such as oxygen. For each glucose molecule that is catabolized from glycolysis through the TCA cycle, 10 NADH and 2 FADH2 molecules are produced. Each of these molecules contains energy that originated from glucose. Electron transport systems present in the membrane transport the electrons to oxygen, forming water, and in the process convert the energy into electrochemical gradients across the membrane. As the electrons are transferred within the ETS pumps hydrogen ions, also called protons, across the bacterial cell membrane. As a result, a gradient forms across the membrane, with a higher proton concentration outside than inside. In this way, the energy of the electrons from glucose is transferred to the energy of a proton gradient. The hydrogen ions will tend to move down their concentration gradient back into the cell, and they do so through a complex called ATP synthase. This flow of hydrogen ions through ATP synthase drives the formation of ATP from ADP, an inorganic phosphate. The production of ATP using the hydrogen ion gradient from the electron transport system and the reduction of oxygen is called oxidative phosphorylation. How does the electron transport system operate? An electron transport system consists of a series of electron carriers that sequentially transfer electrons to the carrier with the next higher reduction potential, that is, to a stronger electron acceptor. Electron flow begins with an initial electron donor, such as NADH, from glucose catabolism. Ultimately, all electrons are transferred to the strongest electron acceptor, which in this example of aerobic respiration is molecular oxygen. A typical bacterial electron transport system used in respiration includes at least 3 functional components, beginning with a complex called NADH dehydrogenase. NADH dehydrogenase then transfers the 2 electrons onto the second functional component, a quinone, indicated Q. The quinone also picks up 2 hydrogen ions from the cell to balance the charge, generating quinol, QH2. The redox step from NADH through NADH dehydrogenase to quinol yields enough energy to pump 4 hydrogen ions across the membrane. Quinones and quinols diffuse within the membrane. Each quinol reduced by NADH dehydrogenase can transfer its 2 electrons onto the next ETS complex, cytochrome BO oxidoreductase. As part of the electron transfer event, cytochrome BO oxidoreductase pumps more protons. This terminal oxidoreductase now transfers the 2 electrons onto molecular oxygen. Oxygen has the strongest affinity for electrons of any other component of the electron transport system. In order to become fully reduced, molecular oxygen requires the electrons from an additional NADH molecule. In total, for every 2 NADH molecules oxidized, 16 protons are pumped across the membrane. The final 2 electrons join with oxygen and with 4 protons from the cell, producing 2 molecules of water.
The chemiosmotic hypothesis explains oxidative phosphorylation. As protons move outward from the mitochondrial matrix or across the cytoplasmic membrane, electrons are transported down the electron transport chain. Proton expulsion during electron transport results in formation of a concentration gradient of protons and a charge gradient across the membrane. The combined chemical and electrochemical potential make up the proton motive force based on this difference in charge and ion concentration. Diffusion of protons back across the membrane, that is down the gradient, will drive the formation of ATP. A molecule known as ATP synthase, which is embedded in the membrane, uses this proton motive force to synthesize ATP. As the hydrogen ions are pumped through the ATP synthase, ATP molecules are synthesized. The hydrogens will then bind to oxygen, forming metabolic water. Take a step back and look at an overview of the entire process of cellular respiration.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Obligate aerobes are organisms that grow only using oxygen as a terminal electron acceptor in cellular respiration. This includes animals, plants, and many bacteria. However, other prokaryotes use a wide range of terminal electron acceptors, including metals, oxidized ions of nitrogen and sulfur. This anaerobic respiration generally occurs in environments where oxygen is scarce, such as wetland soil or in the depths of the human digestive tract. Anaerobic respiration is unique to prokaryotes. They usually possess alternative electron donors and electron acceptors. Respiration using oxidized forms of nitrogen is widespread among bacteria and archaea. Reduction of oxidized states of nitrogen for energy yield is called dissimilatory denitrification. With this, nitrate is successively reduced from nitrate to nitrite
then perhaps on to nitric oxide, nitrous oxide, and eventually nitrogen gas. Dissimilatory nitrate reduction uses nitrate as a terminal electron acceptor, making it unavailable to a cell for assimilation or uptake. Denitrification is the reduction of nitrate to nitrogen gas. In general, any given species can carry out only one or two of the transformations in the series we just discussed. Anaerobic respiration yields less energy than aerobic respiration because the reduction potential of the electron acceptor is less positive than it is of oxygen.